Welcome back to the deep dive. Our mission today, well, it's tackling a really big bottleneck in medical science. We're looking at how we can find safe, effective drugs much faster, drugs that actually extend our healthy lifespan.
It's a huge hurdle, yeah. Anti-aging drug discovery.
Huh.
It's kind of stuck. You've got cell-based systems, the in vitro stuff, but they're just too simple, you know? They don't capture the whole picture of a living organism.
Right, the complexity isn't there.
Exactly. But then you jump to, say, mice, and suddenly things get really slow. We're talking months, even years for experiments, and they're expensive, difficult to manage.
So you need speed, but you also need something that actually reflects, well, a whole aging animal.
Precisely. Biological relevance is key. Which brings us to our main subject today, this tiny nematode worm.
Ah, yes. The source material focuses heavily on Cantorhabditis elegans, C. elegans. Seems like this little worm might hold the key.
It's become a powerhouse for this kind of research. Why? Well, first, it ages super fast. We're talking its entire lifespan plays out in just a few weeks. That's a massive speed advantage right there.
Weeks instead of years.
Right. And second, it's surprisingly relevant to us. About 83% of its proteins have similar structures, homologs, to human proteins. So you're studying a whole aging organism quickly, and it has genetic ties to human biology.
Okay, but the research really stresses something important here. It's not just about lifespan. Why is just measuring how long the worm lives? Why isn't that enough anymore?
That's a critical point, because living longer isn't the goal if those extra years are spent frail or sick, right? We need to measure health span, how long the organism stays healthy and active.
Extending health, not just existence.
Exactly. Think about it. Some known genetic mutations that make worms live longer, like in the insulin signaling pathway, they sometimes come with, well, downsides, trade-offs. Things like slower development early on, or they can't reproduce as well, lower fecundity.
Ah, I see. So just measuring alive or dead at the end might completely miss that the worm was actually less healthy for a large chunk of its life.
Precisely. You might pick a drug that extends life, but introduces some serious quality of life problems.
Yeah.
So we need to track functional health over time.
Which logically leads us to the solution proposed in the research, this new tech called the WormGazer. It automates health span measurement by watching how the worms move. Okay, so why focus on movement? What makes worm locomotion such a good indicator of health span?
Well, think about aging generally, even in humans. A decline in physical activity, in mobility, it's a classic sign. It's the same in C. elegans. Losing that vigorous movement, that's a major feature of their aging process. And crucially, most of this functional decline happens pretty early, like in the first week of their adulthood.
The first week. So you can see the aging effects quite quickly then.
Very quickly. If you can accurately measure how fast that movement declines, and importantly, how a potential drug slows that decline, you've basically captured its effect on health span.
And this is where the speed really comes in. The WormGazer system can apparently assess a compound's effect in just 7 days.
7 days of imaging.
Yeah.
From early adulthood through that main period of decline. It's a game changer.
Because the old way? The source mentioned a comparison.
Right. They ran the traditional manual lifespan assay side by side.
Yeah.
That took 40 days to get the basic survival data.
40 days versus 7. Wow. Okay, you have to describe this manual method because it sounds intense. Checking worms every other day.
Yeah.
And prodding them.
Yeah. With a platinum wire, no less. You have to gently poke any worm that's not moving to see if it flinches. That's how you confirm death.
Oh my goodness. Imagine doing that for hundreds, maybe thousands of worms across dozens of dishes for over a month.
It's incredibly labor-intensive, prone to error too, I'd imagine. And the data you get is just binary. Alive or dead, that's it.
So the WormGazer completely changes that. It's not just alive dead, it's collecting detailed data on how they're living automatically.
Exactly. It's continuous monitoring, no prodding, no disturbing them. And it gives you these really useful metrics, like the fraction of worms moving, so what percentage of the population is active, and the mean speed of moving worms. How fast are the active ones actually going?
So you get population activity and individual vigor. That's much richer data.
Much richer. It's a proper look at functional health, tracked constantly. And the way they designed it is pretty smart. It tackles scalability and keeping things consistent, which are huge issues in aging research.
How so? What makes it different from other automated systems?
Well, a lot of other systems use things like microfluidic chips, or they keep the worms in liquid. That can actually stress the worms out, change their physiology. The WormGazer avoids that. It uses standard lab equipment, the normal agar Petri dishes with the bacteria they eat.
Okay, so it's the environment the worms are used to. That makes sense. But how do they manage to watch lots of dishes at once without some giant, complex robot?
That's the clever part. It's a distributed system. Instead of one expensive high-end camera moving around, they use an array of simpler low-cost cameras. Each camera watches one or a few dishes and is hooked up to its own little computer, like a Raspberry Pi or something similar.
Ah, so you can just add more camera computer units to scale up?
Exactly. It makes it modular and allows for simultaneous parallel imaging. You're watching many experiments happen at the exact same time under the same conditions.
And the source mentioned no moving parts. Why is that such a big deal?
Oh, it's crucial for long-term studies like this. Moving parts mean vibration, potential friction, maybe even slight heat changes. All tiny things that can introduce variability, noise into the data. Aging is sensitive. You want conditions rock solid.
Consistency is king.
Absolutely. And then they nailed that. No moving parts, plus integrated temperature control that's incredibly precise, holds it within plus or minus 5.1 degrees Celsius. Even tiny temperature shifts can mess with worm metabolism or dry out the agar. This system locks it down.
Okay, so stable environment, parallel imaging. How does it actually see the movement if the camera isn't moving or tracking individual worms?
It's elegant, really.
Yeah.
It doesn't need to track Fred the worm. It takes a burst of images very quickly, like 200 images in about 2.5 minutes every 5 minutes.
A little time-lapse burst?
Sort of. Then the software just compares consecutive images in that burst. Pixels that change value, that's where a worm moved. It highlights the path. They set a minimum speed threshold, about 10 micrometers per second, just to filter out any background noise or tiny vibrations.
I see. So you're not tracking individuals, you're getting a population-level measure of total movement and speed.
Precisely. It's similar in principle to the manual lifespan assay, where you rely on population numbers to get a statistically significant survival curve. Here you get a population-level functional curve. It works beautifully for seeing the overall aging trend.
All right, let's get into the validation. This is where the system really shows its power, right? Not just confirming things, but finding those hidden trade-offs. They started with known genetic mutants.
Yes, the classic ones. First up, the age 1 mutant. These guys are known to live longer than normal wild-type worms.
And did the WormGazer pick that up in terms of movement?
It did, yeah. Later in life, exactly as you'd expect.
Mhm.
Between day 2 and day 7 of adulthood, the age 1 worms spent, get this, 93.5% more time moving compared to the normal worms. Clear extended health span.
Okay, so it confirmed the known effect. But then came the interesting part, the trade-off.
This is where it gets really insightful. While they were more active later, the system revealed a cost early on. In that initial period, days 0.5 to 2, the age 1 mutants actually had a lower fraction, moving about 12.1% lower than normal worms.
So less active right at the start of adulthood?
Yeah, and more specifically, if you looked at their speed on day 2, their average speed was down by 27%. So they live longer overall, but they seem to start slower, less vigorously. A real functional deficit early in life.
Wow. That's something a simple lifespan assay would totally miss. You just see the long life at the end.
Exactly. If a drug caused that kind of early slowdown, the WormGazer flags it immediately. It's a potential warning sign about early life health impact.
And they also looked at a short-lived mutant, DAF16.
They did. And again, it matched expectations, but with functional detail. Those worms showed a much faster decline in movement, spending about 23.2% less time moving than the wild type over the first 12 days or so. So the system accurately captured both long and short health spans, quickly and with a contextual nuance.
Okay, moving from genetics to actual drugs. They tested sulfamethoxazole, SMX. That's known to extend lifespan, right? Something about the worm's gut bacteria.
That's the one. And yes, the WormGazer results mirrored the lifespan results. It showed improved health span by day 7, which matched the 40-day survival data. Faster confirmation.
But did it pick up any nuances there too?
It did. Another caution flag. Even though overall movement improved across the experiment, the system detected that around day 2.5, the average speed of the worms on SMX was actually significantly slower than the controls.
So again, an early negative effect on vigor, even if the overall outcome was positive later.
Seems like it. You get the longevity benefit, perhaps, but maybe at the cost of some early sluggishness. The system reveals that complexity.
Fascinating. Then there was the alpha-ketoglutarate, alpha-KG study. This sounds like a perfect case study for why rapid functional screening is so vital.
Oh, absolutely. Alpha-KGG is another compound people are interested in for longevity. They used WormGazer to quickly test different doses.
Dose finding is critical.
Super critical. They found that, yeah, 2 millimolar and 4 millimolar concentrations significantly boosted movement by about 10–12% in that first week. Good result.
But there's a but, isn't there?
There is
Ah.
a concentration that had been previously reported in other studies as effective, 8 millimolar. The WormGazer showed it actually reduced movement, made the worms less active.
So a higher dose, which might have looked okay or even good in a slow lifespan-only study, was actually detrimental to their functional health.
That's what the data strongly suggests. If you'd only looked at survival over 40 days, you might have missed that 8 millimolar makes them less healthy day to day. You might have pushed forward with a suboptimal, maybe even harmful dose.
That really highlights the danger of just looking at lifespan. You need the functional picture, and you need it fast to guide the research properly.
Exactly. This system let them find the truly beneficial dose range and identify a potentially problematic one all within a week. That prevents wasting time and resources going down the wrong path. Hashtag outro. So if you zoom out, what this tech really does is break down those barriers in aging research. You get rapid, standardized, in vivo, and a living animal data. And crucially, it's focused on health span. It lets researchers find compounds that work, find the right dose, and see any negative side effects or trade-offs almost immediately.
Which means, for you, the listener, the whole process of finding drugs that could genuinely help us live healthier for longer, well, that process just got a significant speed boost. The path to finding safe, functional longevity treatments is getting clearer, faster.
Yeah, the potential is huge. And it makes you think, if this system is so good at measuring general health and movement non-invasively, what else could it do?
You mean beyond just aging drugs?
Right. The source material hints at this. Could you use it for rapid toxicity screening? Or maybe adapt it to study other behaviors, things like chemotaxis, how worms
Yeah.
react to chemical signals in their environment. If you can get reliable behavioral or toxicity data in just a week on a whole organism, that could accelerate discovery in a whole lot of areas, not just aging. It's a really versatile platform.